This next unit is just chapter 9, which is muscle. So as we learned in our tissue unit, there are 3 types of muscle tissue in the muscular system. There's the skeletal muscle, which is attached to bone. Remember, that is voluntary, striated, multinuclear, very long and cylindrical. Cardiac muscle makes up most of the wall of the heart. It's involuntary, striated, has intercalated discs, is branched. And smooth muscle, I just lied, didn't I? No, I'm right. Skeletal muscle is long, cylinder. Cardiac muscle, branch. Smooth muscle found in internal organs, involuntary, no striations, and has tapered ends. So we are going to focus the majority of this lesson on skeletal muscle. We will look at some commonalities and differences between the 3, other than what we already know. There are over 600 skeletal muscles in the body. That's the bad news. We just finished bone and we learned quite a bit of the bones. We are not going to learn 600 skeletal muscles. So take a deep breath and be thankful. They are attached to bones and skin of the face to help make expression. They are voluntary, and each muscle is the organ of the muscular system. They are made of skeletal muscle tissue, the nervous tissue, blood, and other connective tissues. So first we want to talk about the coverings that are found on skeletal muscle. And you've probably heard of fascia. These are thin connective tissue around the muscle, kind of like a packaging, if you will. The tendon is connective tissue that connects muscle to bone. In the skeletal unit, we looked at ligaments, that's connecting bone to other bone. Tendons connect muscle to bone. And aponeurosis are sheet-like masses of connective tissue that connects muscle to bone or skin or even another muscle. And we'll only see a few of these in the skull or on the skull and the abdomen. So, if you will, pretend like the muscle is a present and the fascia is the wrapping paper. So you're going to take that off and you have a box. And the box is going to be made of epimysium. It's surrounding the whole muscle. And it's right beneath that wrapping paper, the fascia. What we're going to find is the muscle is pretty complex in its organization. The units of muscle get smaller and smaller and smaller, and they're all kind of the same shape. So that's why I'm using this analogy of a present. So once we open up the whole muscle, we're going to find smaller structures inside, and they will be wrapped in boxes called perimysium. And after we open the perimysium and look inside, there will be smaller structures, and those will be wrapped in endomysium, and that is going to be the cell. So epimysium surrounds the whole muscle. When you open up the organ, the muscle, you're going to be seeing tissues called fascicles. Those fascicles are surrounded by perimysium, and then you open up those tissues and you have the cells, the muscle fibers, and they're surrounded by endomysium. Notice these prefixes. Epi on the very outside, peri around, endo the very inside. This is the lowest level of connective tissue. Because once we go under the endomysium, we're at a cellular level. And so you can't have tissues inside cells because cells are smaller than tissue. So real quick, tendons and then the aponeuroses. So here you see the sheet-like connective tissue. I told you you'd see some on the skull and the abdomen. And then tendons are connecting the muscle to the bone. So this is what I was talking about, that it's kind of tricky. And I will say this is where my students struggle. We practice in class lecture. I have a lot of like card sorts and activities. We practice the heck out of this. because it gets a little bit confusing. So here's that fascia, the wrapping paper, and here's our first box, and it's covered with epimysium. And when you open up the epimysium, it's wrapped around the muscle, okay? And then inside the muscle, you have a tissue. This is the tissue. It's a group of cells working together, and this is a fascicle. All right, so this whole thing is a fascicle, and it's wrapped in perimysium. So if you look at this organ, the muscle, you can see 1, 2, 3, 4, 5, 6, 7, 8 fascicles. Now we're zooming in on the fascicle surrounded by perimysium, and inside these are individual cells. So 1, 2, 3, 4, 5, 6, 7, 8, 9 cells. I'm not counting them because you need to know how many are in there. I'm counting them so you can recognize that this whole thing is one cell. And this is a group of cells called a tissue. And this is a group of tissues called an organ, muscle. All right, so the fascicle is the tissue. And then the individual cells are called muscle fibers. So you can even see the nucleus in some of these muscle fibers. Now, if we look at one cell, one muscle fibers, which it's surrounded by endomysium, you'll see organelles inside. A few shown here are the sarcoplasmic reticulum, a specific type of endoplasmic reticulum, or ER, that's only found in muscle. And these what's called muscle fibrils. Woo. Myofibrils are in the muscle fiber. Muscle fiber is cell. Make a note. This is the cellular level. Myofibril is an organelle, and it's the cytoskeleton made of filaments. And so here are the filaments. There's a thin one and a thick one, and we'll name those later. There's a nucleus in here, right inside the endomysium. There's going to be mitochondria in here. And so this is one cell, and this is looking inside the cell. This is an organelle and looking inside the organelle. Whew, okay. So the space containing the group of muscles and the blood muscles all enclosed by the fascia. And then you can have compartment syndrome where fluid accumulates within that compartment, and it could increase the pressure, and it's going to be severe pain. Remember that the fascia is the portion surrounding the muscle, and it connects to the subcutaneous fascia, which is right under the skin, or to serous membranes. So let's look at the cell. Muscle fiber is what they call the cell. It is multinucleated. We already know that. It's going to have what's called the sarcolemma. That's the fancy term for the cell membrane of the muscle fiber. It is the phospholipid bilayer. The sarcoplasm is the cytoplasm. So what do you notice? Sarco. That means muscle. Now it's going to have a lot of myofibrils, those organelles inside that are the cytoskeleton. They are long. We saw how they run parallel, and they consist of smaller filaments, the thin and the thick. The thin are actin, and the thick are myosin. I remember this because my sin is like overeating, and that's why I'm thick. It just helps for you remember, okay? You don't have to be thick to use my analogy. Sarcomere. Mere is a part. We saw monomer and polymer, and now we see sarcomere. So this is a unit of muscle, and they connect end to end and make up these myofibrils. And I'll show you a picture of this in a minute. The sarcoplasmic reticulum is the ER of the muscle. Its job here is not necessarily to make lipids, but to store calcium. And then what's really unique about the sarcoplasmic reticulum is it has these tubules that are like holes where the sarcoplasmic reticulum goes down deep into the muscle cell. And then a triad is a unit consisting of the one tubule and the two sarcoplasmic reticulum cisternae. Don't worry, you can cross that out. Never going to test you on that. So here is that muscle cell, also called a muscle fiber. It's wrapped in it would be, it's not shown here, but it would be wrapped in endomysium. But here's the cell membrane, and the cell membrane is the sarcolemma, and inside is the cytoplasm called the sarcoplasma. And here are some mitochondria, and here are these big myofibrils. All right. The T-tubule or transverse tubule, I told you it is where a piece of the sarcoplasmic reticulum or sarcolemma, move down deep into the cell. So these are these T-tubules, and they are integrated down near the sarcoplasmic reticulum and amongst these myofibrils. If you look inside the myofibril, you can see these thin myofibrils, myofilaments. Just kidding, this is all a myofibril. Myofilament is the actin, and these thicker pink looking ones are the myosins. And so I told you that the sarcomere is a unit of muscle, so you would go from one end to the other. That is one sarcomere. So this has 1, 2.5 sarcomeres in that one myofibril. Let's see. And there's another nucleus of another cell, of the same cell, because it's multinucleated. All right. So myofibrils are made of sarcomeres that connect end to end, and they have this striated pattern, right? The stripes you can see are made by the arrangement of those myofilaments in the myofibril. So one sarcomere consists of these structures: an I band, an A band, an H band, a H zone, a Z line or Z disc, and an M line. Okay, so the I band is a lighter band, and it's just thin actin filaments. They're thin, which makes it look lighter under the microscope. There's not a lot of stuff there. The A band is the dark band, and it's made of the thick myosin, and it overlaps a little bit with the thin. The H zone is the center of the A band, and it's just the thick filaments, no overlapping. The Z disc is the sarcomere boundary, so it's at the end or the center of an I band. And we'll show you this in just a second. And the M line is the center of the sarcomere and A band, and it anchors the thick filaments. So let's look at this. One sarcomere has all those parts. So this kind of looks like the letter Z, so that's easy to remember, the Z lines, and Z's at the end of the alphabet. So these are the ends of one sarcomere. The A band is the thick filaments, and you can see areas where they overlap with the thin filaments. Where there's just thick filaments and no thin is the H zone and the A band. The M line is right here. A band is all of the myosin. Where there's just actin is the I band, and in the middle of that is the Z line. So this would be the I band, and this would be the A band, and this would be the H zone, and this is the M line. And so you can see here's a Z line and here's a Z line. Look at this A band. You can even see an M line a little bit. So what's going to happen is this sarcomere, when it contracts, these parts are going to move, and the sarcomere is actually going to get smaller. The Z lines are going to come together as these thick filaments pull on the thin filaments. So the thick filaments are myosin protein, and they have little heads on them that form what's called cross bridges when they connect with the thin filament. The thin filament structurally is more complex. It is also protein called actin, and it has troponin and tropomyosin on it. And those two also proteins block the cross-bridging formation of the heads on the thick filament. So here is the myosin protein, the thicker protein, and here are these heads that are coming off of the myosin. It has billions of them. And then your thinner filament, so these like spheres or whatever, that's the actual actin molecule. And then you've got your tropomyosin that's blocking. I don't know if you can see, but there it looks like it reminds me of like snap beads, and there's a little hole right there. There's a little hole right there, and there's a little hole right there. Well, those are the binding sites for these myosin heads. But notice the tropomyosin is blocking those. So these heads can't connect with the binding sites because they're blocked by this tropomyosin. This blue structure is the troponin, and when it changes shape, you can see it's kind of connected to the tropomyosin. When it changes shape, it slides the tropomyosin out of the way, and that's going to allow these heads to connect, forming a cross bridge. So to contract a skeletal muscle fiber, it requires a lot of interactions. I tell it as a story in class, and we draw this out, just telling the story and practicing. And then we put all they actually write their own notes because they know the story. But what's going to happen is, like I said, those myofibrils are going to interact and slide past one another. This is called the sliding filament model. And the sarcomere, those Z lines come together and shortens the sarcomere. And as that one muscle fiber shortens, so do other muscle fibers, and the whole muscle cell organ shortens and pulls on the insertion towards the origin. So we need to talk about the neuromuscular junction. This is a synapse. A synapse is where a neuron is interacting with another cell. And because it's interacting with a muscle cell, we call it a neuromuscular junction or a myoneural junction. And this is where the axon and motor of a motor neuron interacts with the muscle cell fiber, the cell of the muscle. And skeletal muscle fibers do only talk to motor neurons. Motor sounds like movement, so they're going to help move the muscle. So there's a few parts of the neuromuscular junction. There's the motor neuron that's going to send the information to the muscle cell to contract. There's the motor end plate, which is a folded portion of the skeletal muscle fiber where it can bind to neurotransmitters released by the motor neuron. The synaptic cleft is the space between the neuron and the muscle, and the transmitter released by the motor neuron has to travel across to the motor end plate. The synaptic vesicles hold the neurotransmitter in the motor neuron, and then the neurotransmitters are the communicative chemicals that are released by the motor neuron and talk to the muscle fiber at the motor end plate. So here's this motor neuron, and so what you're doing is you've thought to move something, or you're having a response to some stimulus and you need to move your muscle. Maybe you just got burnt and you need to contract some muscles to move your hand off the hot item. And so that information is coming down from the spinal cord or brain, and it's coming here. It's called an action potential, this information, electrical stimulus, and it's coming here to the axon terminals of this motor neuron. And so we're at the axon terminal, and in that terminal are these little synaptic vesicles, and they're holding the neurotransmitter. Our specific neurotransmitter is acetylcholine. And so what's going to happen is once an action potential reaches this axon terminal, calcium's going to come into this cell, into the neuron. This is nervous tissue, or a nervous cell, right? And that calcium coming in causes these vesicles to do exocytosis and release the neurotransmitter acetylcholine into this synaptic cleft. They'll diffuse and they'll bind to this space called the motor end plate. And what's going to be found there are little receptors that can receive this information and cause the response of contraction of this muscle fiber. So acetylcholine is the specific neurotransmitter, and the nerve impulse called an action potential is what causes ACH or acetylcholine to be released. And what does that, as I mentioned, is sodium, specifically, we'll just focus on sodium, entering the sodium, is going to also enter the muscle, and potassium will move out, and that's going to create an action potential on the membrane of the muscle. So, an action potential, we'll learn more about that in our next unit, but just know that an action potential is this wave of energy. It moves down here, causes calcium to move into the neuron. That causes the synaptic vesicles to release neurotransmitter. Neurotransmitter diffuses across the synaptic cleft, binds with receptors on the sarcolemma, and that causes calcium and potassium to be permeable and causes another action potential on the sarcolemma. That sarcolemma has the action potential move as it goes down into a T-tubule and will eventually release calcium ions from the sarcoplasmic reticulum. So there are muscle disorders like autoimmune disorders where the antibodies actually attack the receptors, and the person only has about a third of the normal number, which leads to muscle weakness and fatigue. And there are some treatments for it. Muscular dystrophy is another one where we have a scarcity of a protein, and that binds the internal side of the muscle cell membranes and holds them together. And so if you don't have this, the muscle cells actually die. Clostridium botulinum is an anaerobic bacteria you get from food poisoning. This is why you don't buy cans that have been dented in. Actually, a lot of stores will sell those at a discount because they could have micro fissures in that can where bacteria can get inside. There's not air in there, and so it can grow well and make this toxin. And so this is where Botox is extracted and used to what Clostridium botulinum. When you ingest this toxin or have this toxin in you, it paralyzes the muscle. And so that's how Botox is used, to paralyze the muscles of the forehead so that you can't raise your eyebrows as much and you don't create wrinkles. Okay, let me see. So I told you that the action potential is going to move down a T-tubule, and that's going to cause calcium ions that are stored in the sarcoplasmic reticulum to be released. What's going to happen is these calcium ions are going to interact with the troponin and the tropomyosin complex. When the calcium binds to the troponin, it changes shape, which moves the tropomyosin off of those binding sites, and then the myosin heads can bind to actin, forming a cross bridge. So let me back up to this. I wish there was a better picture. Let me see if there is. There's not a great picture, but your lab activity will help you. So let me go. Okay. So an action potential is going to be generated on the sarcolemma, and it's going to actually go into these little T-tubules. So the action potential, the wave of energy, runs down the sarcolemma and down into this T-tubule. And when it interacts with the sarcoplasmic reticulum, the calcium is released. And the calcium is going to diffuse down here and interact with these thin actins, where they move they change the shape of the troponin, and that moves the tropomyosin off of the binding sites so that the heads can interact. And that's how you form a cross bridge. So that's where we are here. So once calcium is available, you can see the calcium is binding to the troponin, and it's slid the tropomyosin off of the binding sites. Here's those myosin heads. And what they're able to do now, because those binding sites are available, is attach to them. When these myosin heads attach, they change shape, and it's a bend backwards. It's kind of like pulling on I think about a tug of war where all your friends grab hold ahead of you, and then once you grab hold, you change shape, you bend your arm, and it pulls the thin rope towards you the same way the myosin is pulling the thin actin towards it. Now, what's interesting is we need ATP to bind to the myosin heads for them to let go and reach and do this again. And so this can continue in a cycle as you continue to contract your muscles more and more, but you also need ATP to release the myosin heads if you want to relax the muscle. So ATP, while it's needed, make a note, ATP is needed to release myosin heads from those binding sites, which is contrary to what most people think. Think about when somebody dies and they're no longer able to provide ATP, the energy molecule, to release their myosin heads. That's what causes the rigor mortis. So like I mentioned, this is called the sliding filament model, where those sarcomeres shorten as the thick and thin filaments slide past each other. The H zone and the IO band will narrow, the Z lines move closer. The thin and thick filaments never change shape. They're just overlapping, sliding past each other. And so let's look at that. As here, notice there's no calcium in the area, so the myosin binding sites are covered by the tropomyosin. But once calcium is in the area, it binds with the troponin, which moves the tropomyosin over and exposes the binding sites. When the myosin head grabbed hold and moved, look what happened. The Z lines were out here, and they moved in a little bit. And we can continue to do this as long as there's ATP and contract it further. And look how much shorter the sarcomere is than when we started. Now, the myosin did not change shape or size. The actin did not change shape or size. We're just sliding them as we contract this muscle. So the myosin head attaches to the actin binding site, forming that cross bridge, and then it pulls the thin filament towards the center of the sarcomere, towards that M line. ADP and phosphate are released from myosin head, so you have to have a new ATP molecule to bind to myosin to undo the linkage. So the cross bridge breaks. ATP splits apart when that happens, and then the myosin heads can go back to the original position, ready to bind again, forming another cross bridge. And so we saw that happening. So how do you relax? A few things, right? We know we're going to have ATP to undo the myosin heads from the binding sites, but you've got to go to the beginning. What caused all this contraction to occur? It was the release of that acetylcholine in the synaptic cleft that caused this chain of reaction. So acetylcholinesterase is an enzyme. Remember, anything that ends in ACE is an enzyme. And it decomposes the acetylcholine that's found in the synapse so that it won't keep this contraction going. The muscle impulse stops when that acetylcholine is decomposed. And there's no longer an impulse going down the T-tubule into the sarcoplasmic reticulum. So calcium's not being released anymore. In fact, calcium pumps pump calcium back into the sarcoplasmic reticulum. Because we're using a pump, we're going to need ATP. So this is why energy is required to actually relax a muscle as well. Those troponin and tropomyosin complex go back to covering the sites, and the myosin and actin binding are now prevented, and your muscle fiber can't contract, so it relaxes. So where do we get ATP for all this contraction? Well, there's already ATP in the cell, and we use those up really quickly. It's a tiny, tiny amount. You also have creatine phosphate and in small amounts in the cell already. And what happens is creatine phosphate can quickly convert ADP to ATP. And you get about 10 seconds of muscle contraction for that. And then you have to rely on cell respiration, where the mitochondria uses oxygen and sugar from your food to produce mass amounts of ATP. And so here is showing energy sources for contraction. So it says when cellular ATP is high, you'll use this, and the ATP can actually make creatine phosphate. But when you have low ATP, you can use that creatine phosphate to change ADP very quickly to ATP. This section goes into the phases of cellular respiration, and you will not be tested on cellular respiration, so please X those out. But I do want you to know about oxygen debt. When you're at rest or you're doing like easy exercise, you're able to breathe easy, your cardiovascular systems have a constant supply of oxygen to support aerobic respiration, you're good. But in anaerobic threshold, which is lactic acid threshold, you have to shift your metabolism from aerobic to anaerobic. This happens during strenuous activities where you're not supplying enough oxygen and you produce lactic acid. You know this has happened when you're sore the next day from this anaerobic metabolism. And so most of the oxygen in your strenuous exercise is used to produce ATP, not to convert lactic acid to glucose. So at the end of your strenuous exercise, your muscles may be left with an oxygen debt. And so this is the amount of oxygen needed by your liver cells to convert the lactic acid back to glucose and to restore your muscle ATP and your creatine concentrations. And so this only happens during strenuous exercise, and it's after you've exercised and you're continuing breathing to help your liver change your, to recover pretty much. Muscle fatigue is when you've worked your muscle out so much you can't contract it anymore. It could be because of decreased blood flow. It could be because of ion balances. The loss of desire to continue the exercise, or some people think an accumulation of lactic acid. But think about muscle fatigue I think about like doing drop sets or whatever, and there's a point where you have like no weight hardly and you can't contract the muscle. Muscle cramps are a sustained involuntary muscle contraction, and they think this is caused by the electrolyte concentrations. And so this is why athletes, when they do have muscle cramps, they're given Gatorade or another electrolyte drink. I can't think of any others right now, but you probably are thinking of some. Remember what electrolytes are. They're ions. So they're sodium, potassium. Didn't we need those for this process? Calcium ions. Some people drink pickle juice because it has a lot of ions in it. Mustard, those kinds of things. So if I know as a pregnant woman in the middle of the night, I'd get these contractions because my baby was still in my electrolytes. So I would go drink pickle juice. Heat is a byproduct of cell respiration in active cells. That's why when you're working out, you get hot. In fact, muscle cells are the major source of your body heat. More than half of the energy released in cell respiration is actually heat and not ATP. All right, so muscle contraction can be observed by removing a single skeletal muscle fiber, one cell, and connecting it to a device that records these changes in a muscle fiber. And this is actually a lab you're going to do. So I'm not going to focus too much on it because you'll learn it in your lab. But the threshold stimulus is the minimum strength of a stimulation that a muscle fiber needs to cause a contraction. When it reaches threshold, the action potential is generated and you end up doing all the, once you have that action potential that goes down the T-tube, you'll do all the steps to end up making those cross bridges. One action potential from a motor neuron releases enough acetylcholine to produce the stimulus of a muscle fiber. Now, a twitch is the contractile response of a single muscle fiber to a single impulse. And there are periods of time associated with the twitch. There's a latent period, which is this delay between the stimulation and the actual contraction of the muscle. There's the period of contraction and the period of relaxation. So pretty easy. You stimulate right here and you have this pause where it takes some time for the cross bridges to form, and then you have this contraction and then you start to relax, and there goes the twitch. So the length tension relationship, the length of the muscle fiber before it stimulates determines the amount of force it can develop. So the optimum starting length is resting, and this allows the greatest force to develop. So the stretched muscle fibers develop less force since some of the myosin heads can't reach those binding sites. So the best way to have the strongest muscle is not to to stretch it, but to just go from rest. The shortened muscle fibers also develop less force since the sarcomeres can't shorten any further. Like once you're contracted, it's harder to contract even more. And so they're saying that like this is your optimum length for developing force. If you are already shortened or you're stretched out, you're not going to be able to obtain the same amount of force as a resting muscle. Now, summation, sum means to add. So this is where the force of individual muscle fiber twitches can actually add together to produce a sustained contraction. And then tetanus is where you have a bunch of twitches that become sustained contraction. Now, complete tetany occurs at very high frequencies of stimulation. It does not occur in the body. We can only see this in the lab. But this is showing these little twitches. And when you get them close together, they can actually add. And if you have a high frequency of them, you can release tetanus. And so a motor unit is a motor neuron plus all the muscle fibers it controls. So a whole muscle consists of many motor units, so many neurons talking to different muscle fibers. And then recruitment is the increase in the number of motor units activated to produce more force. And again, you'll see this is pretty easy. You have here two motor units. Motor unit 1, if you look, is talking to many muscle fibers. And then motor unit 2 is talking to different muscle fibers, and so you can engage one or both of these for different strengths in the muscle. So the smaller motor units usually recruit first, and then the larger motor units are recruited later, and the summation and recruitment produces these sustained contractions. Muscle tone is a continuous state of a partial contraction, and so that's when you see somebody whose tone, their muscles are at rest, but they're in a continuous state of partial contraction. And the last thing we're going to learn in this lecture is types of contractions. There's isotonic contractions and isometric. We've seen iso before, it means same. So if you'll think about isotonic means same force, this means same length. This one's easier for me to remember for some reason. I use a meter stick, or metric means length. Okay, so if the muscle is contracting but it doesn't change length, it is an isometric contraction. And so the tension is developing but the muscles aren't moving and the bones aren't moving. So how do you do these types of contractions? Well, if you've ever done a wall sit or a plank or done yoga stance, you are definitely working those muscles. They are contracted, but you're not in motion. Isotonic is where the muscle contracts and changes length, and you have motion. So there are two types of isotonic contractions. There's concentric and eccentric. Concentric is when you're shortening your contraction, like you're lifting an object, you're doing a bicep curl. Eccentric is the lengthening of that muscle contraction. And so it's when you're allowing the object to go back down. And so both of these are working the muscles in different ways in what's called an isotonic contraction. So here are, would be a concentric. We're doing our bicep curl. If we're moving this down, it's an eccentric. And if you're holding the bar and you're just sustaining that contraction, that would be an isometric. So these are both isotonic. I just lost my thought. And then this is the isometric. And remember, in isotonic, we have concentric and eccentric contractions. All right, when we come back, we'll talk about fast and slow twitch muscles. We'll look at smooth muscle and cardiac muscle, and then we'll start naming the muscles of the body that you need to know. For this unit, by the way, 2 lectures and then 3 labs and a pretty lengthy practice, because I really want you to practice these muscles and all the information here. Thank goodness we're only doing 1 chapter, right?